So both of these particular superbugs, obviously they resist a specific antibiotic, which is the antibiotic that's listed in its name. So we have to figure out different treatment options to be used to treat these particular infections. So the choices we can't use are methicillin and we can't use vancomycin. So that's another reason why we have so many different strategies that are employed in terms of when we develop our antibiotics. So we just target different aspects of the bacterial cell and hopefully try to get that to work and hope that these bacteria don't resist the new mechanisms that are used to try to treat them. Some other superbugs that express resistance are multi-drug resistant Mycobacterium tuberculosis. Remember we talked about Mycobacterium tuberculosis being treatable through the use of a combination of different drugs. And so what they're finding is that Mycobacterium tuberculosis is not being impacted by the multi-drugs that are being thrown at it. So again, using research to try to figure out different mechanisms through which we can target this particular species of bacteria. There's also multi-drug resistant strains of Acinetobacter, Burkholderia, E. coli, Klebsiella, Pseudomonas, Stenotrophomonas, Salmonella, Shigella, and Neisseria gonorrhea. All of these organisms we've been treating for years and years and years, and they've been able to develop their own resistance against the treatments that we've thrown at them. Because of that, we're constantly working to figure out what's better treatment options for patients infected by these organisms. There's also a species known as Streptococcus pneumoniae, which produces an enzyme called beta-lactamase. And beta-lactamase is an enzyme that can break down the beta-lactam ring structure that's found in a lot of cell wall protein or cell wall synthesis inhibitors such as penicillin. So because the enzyme is produced, the penicillin is ineffective because the penicillin uses a beta-lactam ring structure to destroy the peptidoglycan of the bacterial cell. But if the bacteria expresses this beta-lactamase, then obviously the treatment isn't going to work. So Streptococcus pneumoniae is an organism that is able to secrete this enzyme and combat the treatment that we're using like a penicillin. Some other superbugs are Haemophilus influenzae, and then Klebsiella pneumoniae, which is a carbapenemase producing organism. So how do bacteria become resistant to drugs? Well, some bacteria are just intrinsically resistant, which means they have a natural resistance and the treatments just aren't going to work on them. The reasons for this is because they lack a specific target site for that drug, so the drug isn't able to employ its attack mechanism. Or the other reason is that the drug is not able to pass through the organism's cell wall or cell membrane. So if the drug can't get to its particular target site and has been blocked, then obviously it's not going to be effective. And so if it's not effective, then we refer to that as resistance, but this is not really inherited resistance. It's more of an intrinsic resistance. It's just the treatment plan didn't work. Acquired resistance is what we're more concerned with, because with acquired resistance, this is when bacteria that originally were not susceptible to the treatment, but then over time they were able to adapt and now they are resistant to the treatment. And so what happens here is you have basically the passing of resistance genes from parent offspring to daughter offspring. And so once you have a buildup of these daughter cells that are resistant, then these recipient cells that have become resistant, then you have multiple cells being resistant to a specific treatment. A drug that is used to target a specific cell has to pass through the cell membrane. So to do this, it will actually have to bind to the surface of the cell as kind of an attachment site. And we refer to these attachment sites as drug binding sites. Now, there are some organisms that can induce a chromosomal mutation, and once that mutation is being expressed in the cell, these drug binding sites can be altered, which will impact and limit the drug from binding to these sites. So if the drug can't hang on to, say, the peptidoglycan or the cell wall, and it can't attach itself, then obviously it's not going to be an effective treatment plan. So this is an example of resistance, and this particular chromosomal mutation can be passed on to the daughter cells of the parent cells, making or increasing the chance of resistance among the bacterial cells. And here is kind of an image showing you how cells become resistant. So this takes place through the process of conjugation, which is where you have two bacterial cells joining up to each other. And we have what's called a plasmid transfer. In this diagram, we're going to talk about three different resistance mechanisms. Through plasmid transfer, what you see is you have conjugation, you have what's called a donor cell binding to a recipient cell, and the donor cell will replicate this plasmid, and you can see where the green is, that's the resistant gene. Once the plasmid's been replicated, a copy of the plasmid is sent through the sex pilus and into the recipient bacterial cell. Once the plasmid is inside of the cell, the resistant gene is now in the cell. And so once this resistant gene gets translated into a protein, it's going to be activated and cause this cell to become resistant. So that's one example of how resistance is acquired. Another example is through the transfer of free DNA, which is also known as transformation, which is something we've talked about in here. And with transformation, this whole bacterial cell will just do an uptake of free-floating DNA. So free-floating DNA is taken up by the cell, and now the DNA is inside of the cell, and the cell can then replicate or transcribe this gene into messenger RNA and then translate it into protein for expression, and that will cause resistance to develop in the cell. And then lastly, the next one is the process of transduction, where you have a bacteriophage, which is a type of virus, and that virus contains the resistance gene, and it is going to attach its site or itself to the cell and then inject its DNA that contains the resistance gene into the cell. And again, you have the process of transcription and translation, and that causes the cell to be resistant. So essentially, you have three mechanisms in which resistance genes are passed. You have conjugation, which is plasmid transfer. You have the uptake of free DNA, which is called transformation. And then you have the transfer of DNA by a virus, which is called transduction. And this is basically everything that I've already talked about. There's a lot of text on this slide. Sorry about that. To enter a bacterial cell, a drug must be able to pass through cell wall, cell membrane. Well, we talked about that. Chromosomal mutations can change the structure of the cell membrane, blocking the drug from attaching or entering the cell. That leads to resistance. Bacteria can develop the ability to produce an enzyme that destroys or inactivates the drug. That's going to be basically resistance. And then many bacteria become resistant to penicillin because they have acquired the gene for penicillinase production through the process of conjugation. So that's simply the passing of a resistance gene from donor cell to recipient cell. And You can also have plasmids being passed through that sex pilus in the process of conjugation. And one thing I didn't say was what's being passed here in this resistance gene is called the R factor, which stands for resistance factor. Okay. Here is just a diagram outlining again those key caveats. The altered target site being changed, so that way the antibiotic enters the system. It's going to change the shape or denature or block whatever target it's going after. It can do the same thing to inhibit different pathways from forming. It can block different enzymes to inactivate metabolic processes. And then you can also have essentially an attempt at penetration. And once you have that, then you have kind of a decreased uptake of the bacterial cell of the antibiotic. So when this takes place, then the bacterial cell starts to become resistant. To prevent target sites from being targeted, what the bacteria will do is it will change the shape of the enzymes or the target, and when that shape is changed, then the antibiotic will no longer fit, and that's what's happening here in this image. And then in this image as well, you simply have enzymatic activity being blocked, which is allowing this bacterial cell to be resistant to the antibiotic. Now, bacteria can also use these specialized pumps that help to transport different types of drugs through the cell membrane to cleanse this bacterial cell of the antibiotic treatment. So this will also lead to resistance among bacteria. And what happens with these multi-drug resistant pumps, also known as MDR transporters or efflux pumps, is that the pumps just simply pump the drug out of the cell before the drug is able to do what it's intended to do, which is to damage or kill the cell. So bacteria can get this resistance to different antimicrobial agents through mutation in the chromosomes, or they can acquire new genes from bacteriophages through the process of transduction. They can also pick up naked free-floating DNA that contains the R factor through the process of transformation, or they can also pass on the R factor through the process of conjugation. And here are these mechanisms that we've talked about, essentially. Basically, you can have a bacterial cell change its structure, or change the structure of the binding sites on its outside to prevent the drug from binding to the cell. The bacterial cells can also change the ability of the cell membrane to allow the passing of the drug through the cell membrane. The bacterial cell can make lots of resistant cells through the process of conjugation, transduction, and transformation. And then when this happens, that allows the drug to be destroyed or to be inactivated. With the production of multi-drug resistance pumps, this is going to help the bacterial cells become resistant because they can pump out the drug from the cell. So, like I said before, beta-lactamases with the Streptococcus pneumoniae is a very good defense mechanism for many bacterial cells. Penicillins and cephalosporins have that beta-lactam ring structure, which I mentioned earlier. And with this particular structure, this enzyme is able to target it and destroy it. And once the beta-lactamase destroys the beta-lactam ring, then that makes the penicillin and cephalosporins inoperable. And So this is a big problem and can lead to resistance and different